Modern large language models rely on self-attention to process information. This mechanism maps the relationship between every single token in a sequence, providing deep reasoning capabilities. But that density comes with a prohibitive computational cost. As the context length grows, the memory and processing requirements scale quadratically. Selective state space models, like Mamba, bypass this bottleneck entirely. They process sequences in linear time by propagating information through a recurrent state update, relying heavily on diagonal transition matrices to keep the math fast and hardware efficient. Self-attention builds a dense web where every data point communicates explicitly. Mamba forces information down parallel, non-intersecting tracks. Because the transition matrix is diagonal, hidden state dimensions evolve in complete isolation. This lack of communication restricts the model's capacity for complex, high-dimensional reasoning. Linear efficiency in standard state space models is a direct function of this diagonal parameterization. High-speed processing is achieved by sacrificing the cross-channel interactions that give transformers their power. The Motif-Mamba architecture proposes a mathematical workaround to this limitation. The goal is to force cross-dimensional interaction within the state space model without triggering the quadratic computational penalty. The solution borrows from biological neural connectomes. In the brain, local circuit organization relies heavily on network motifs. These are recurring, highly stable connectivity patterns between small groups of nodes that govern how neural populations process information. Researchers translated this concept into an artificial sequence model by injecting a compact, structurally constrained pathway directly into the standard Mamba hidden state update loop. Motif-Mamba tests whether we can mathematically embed these biological topological priors into artificial state space dynamics to guide how hidden states interact. The standard discretized Mamba update equation calculates the next hidden state by applying a self-decay term to the previous state and adding the new input. This diagram details the exact insertion point for the Motif-Mamba architecture. In panel A, we see the standard decay and input terms, with a new motif coupling term slotted right in the middle of the state update. If researchers had used a standard, dense recurrent matrix, often denoted as W for this coupling term, it would require calculating interactions across every single state dimension. That specific mathematical operation would destroy the parallel efficiency of the Mamba architecture. To bypass the computational wall, the architects fractured that hypothetical dense matrix into two separate low-rank matrices, Mi and Mj. During the forward pass, matrix Mj takes the high-dimensional hidden state and projects it down into a highly compressed latent space, creating a compact state vector called Zt. The cross-channel interaction happens entirely within this tiny latent bottleneck. Matrix Mi then takes that compressed combined information and projects it back up into the original high-dimensional state space. By doing the heavy lifting in a smaller mathematical space, the computational complexity shifts. Instead of scaling with the square of the state dimension, the recurrent complexity becomes OLN plus OLNR, where R is the tiny low-rank dimension. This low-rank bottleneck mathematically forces the isolated state channels to communicate with one another, preserving the hardware-efficient parallel scan. Simply adding a low-rank pathway isn't enough to improve performance. Without a structural prior, the pathway fails to organize cross-channel interactions effectively during training, yielding almost no measurable gain over the baseline. This image outlines the specific biological priors used to solve the organization problem. Panel C displays different three-node motifs. Motif 2, for example, represents a stable convergent structure where two nodes point to a single target. Others, like Motif 12, incorporate complex cyclic feedback loops. Applying these motifs to a neural network presents a mechanical issue. Identifying motifs requires counting discrete binary connections in a graph, but training neural networks requires continuous differentiable equations to calculate gradients during backpropagation. To bridge this gap, the researchers engineered a continuous relaxation proxy. They substituted the binary graph connections with differentiable functions, allowing them to extract the learned motif frequency from the network matrices and formulate a specific motif loss. As shown in Panel D, the model actively compares its own motif statistics against the target biological distribution. This acts as a structural constraint during backpropagation, optimizing the low-rank factors towards specific target topologies as they update. Continuous relaxation bridges the gap between static graph theory and dynamic neural network optimization. We know the structural constraint is applied during training. The critical analytical question is whether penalizing the model with a graph topology actually alters its internal physics and state evolution during sequence processing. These flow fields visualize the actual physics inside the network. Look at the motif blank graph on the left. Without a biological prior, the projected flow and the empirical trajectories collapse into a tight cluster near the origin. This collapse proves that an unconstrained low-rank pathway fails to induce meaningful cross-channel interaction. The mechanism is present, but the model does not utilize it. Contrast that with the motif-2 and motif-12 flow fields. The latent trajectories exhibit significant structural displacement, driven by highly organized vector arrows with significantly larger flow magnitudes. By imposing a specific topology, the architecture dictates the intensity and organization of the recurrent interactions across the internal channel population. The mathematical structure in the latent space dictates how the model performs externally. We can test this by evaluating the model on the synthetic induction heads memory task, which requires the network to remember and retrieve Q-target associations across a given sequence. This chart tracks accuracy as test sequences scale beyond the initial 256 token training context. The orange line, standard Mamba, suffers rapid, continuous decay immediately after this boundary. The green line, Motif-Mamba, sustains high accuracy at longer contexts, successfully retrieving associations up to 16,384 tokens. The stable, convergent vector fields generated by the biological topology in the latent space directly result in superior long-context memory retention. Motif-Mamba proves that we can safely and mathematically import topological priors from neuroscience to govern the behavior of artificial sequence models. By injecting these constraints through a heavily compressed mathematical bottleneck, the architecture gains complex cross-channel reasoning capabilities without sacrificing its linear time processing speed. This shifts how researchers interact with state space models. Instead of treating recurrent updates as a fully opaque process, enforcing specific network motifs turns the internal state evolution into a structurally analyzable system. As context windows continue to stretch, scaling by throwing raw compute at quadratic bottlenecks becomes unsustainable. The future of efficient artificial intelligence relies just as heavily on structural intelligence derived from biology as it does on raw compute.